Hello and welcome to this webinar titled Just-in-Time Release of CAR T-Cell Therapies, hosted by Biopharma Asia magazine and presented by Irving Ford, Head of CAR T QC Laboratories at Celgene. This presentation will be followed by Smart Quality Solutions for CAR T Product Release and presented by Laurie Dahn, Pharma Microbiology Scientific Director at bioMérieux. My name is Stephen Edwards and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now please allow me to introduce our first presenter. Dr. Irving Ford is currently the Head of CAR T QC Laboratories at Celgene. Prior to joining Celgene, Irving was a significant contributor for activities supporting the commercial approval of Novartis' CAR T product Chimera. Irving has over 28 years of QA/QC experience in the pharmaceutical/biotechnology and cell and gene therapies industries, including but not limited to non-sterile, sterile, and low bioburden manufacturing production. Irving has designed, implemented, and managed paperless laboratories. Irving has a successful track record working with laboratory, facilities, validation, manufacturing, regulatory, QA, R&D, and compliance personnel to manage and resolve complex quality and manufacturing issues. I will now be handing over to our first presenter. Welcome, Irving.
Thank you, Stephen. Hi, as Stephen said, my name is Irving Ford, and I am currently the head of CAR T QC here at Celgene in Summit, New Jersey. And today I'm going to talk about contamination controls testing and release strategy for CAR T products. And first, I have to give a disclaimer that the views and opinions within this presentation are those of the presenter and do not represent the views of Celgene or Celgene's regulatory positions. So before I start, I just want to give you a little history about CAR T manufacturing. So currently there are two products on the market for CAR T manufacturing. They're Novartis' Chimera and Kite's Yescarta. And a little about these products is that this is pretty much the last treatment option for patients. They have typically gone through 3 other options that have failed. So this is pretty much a life or death situation for the patient. So when we say that we put the patients first in CAR T, we really have the life of the patients in our hands. And by the time we get their blood and process it and get it back to them, some patients have even expired between that time. So as you can see, getting products manufactured and back to the patient is very important for the well-being of the patient. So because of that, I've explored holistic approaches to trying to get the product back to the patient in a hurry from a microbial standpoint, because typically the microbial testing takes the longest at the end of the manufacturing process. So I have explored ways of trying to eliminate or reduce those time frames so that we can get the product back to the patient so the patient has a better chance of survival. So today in my presentation, I'm going to give 2 options for release of CAR T products. The first option is just-in-time release, and the second option is a 2-day release. So for those of you who are familiar with parametric release, you may be familiar with terminally sterilized products that you don't have to do any microbial testing at the end because it's terminally sterilized. So that's kind of the equivalent of a just-in-time release. So if you think about parametric release, now we're going to switch the page and talk about just-in-time release for CAR T products. And for those who may be a little afraid of taking the plunge into going into a just-in-time release, I'm also going to provide you with a different option, which is called a 2-day release. So let's get started. So in traditional pharmaceuticals, everybody, when they come to CAR T manufacturing, most people either come from sterile or non-sterile. So they either follow like Annex 1 or they follow the non-sterile guidelines. But for CAR T manufacturing, it's a unique product in that we cannot assure the sterility of the raw material that we're receiving because the raw material is actually the patient's blood itself. And because of that, most CAR T products do not have a label claim of sterile, and as such, we are not subjected to following Annex 1. We actually are subjected to the ATMP guidance document, and that's what we follow. So a lot of CAR T companies are just getting started, so they automatically revert to Annex 1. but because your product is not sterile, nor does it meet the definition, the traditional definition of sterility, we follow the ATMP guidance documents. So in the ATMP guidance documents, of course, it talks about risk, and more risk. So because CAR-T manufacturing doesn't fall into the traditional way of manufacturing, because everything in CAR-T manufacturing as of today is mostly manual aseptic manipulations. However, we are transitioning into more automation, but for right now, when you think of the process for CAR-T manufacturing, you think of human beings. In our process, in our manufacturing process, humans are our machine. So everything is pretty much done by a person. And in order to manufacture in this regards, we have to have a risk-based approach. So I'm going to highlight some of the risk-based directions that are given in Annex 1. So in Annex 1 and 2.13, it says the risk-based approach is applicable to all types of ATMPs, and it applies in an equal fashion to all types of settings. It is highly dependent on the biological characteristics and the origin of the cells and tissues, the vectors, and the transgenes. And I just want to highlight an important thing. It says the level and characteristics of the expressed protein for gene therapy products, the property of other non-cellular components, the raw materials, matrices, and the manufacturing process. So again, here is giving you an idea of what you need to look at from a risk-based standpoint. Remember, for CAR-T manufacturing, the raw material for us, the main raw material, so the main active ingredient is the patient's blood itself. The next risk. And the ATMP guidance document says, consider all the potential risks related to the product or the manufacturing process on the basis of all the information available. So what they're saying is that you have to take into consideration everything that you know about your manufacturing process, and your risk should be based from the incoming materials all the way to the release of your product. And finally, I'm just going to take a subset of this. The controlled mitigation measures should be based on current scientific knowledge and the accumulated experience. And it says, ultimately, this evaluation is linked to the protection of the patient. So once again, keep in mind that for CAR-T, our patients are super, super sick. They're actually at the point of death. This is the last treatment option for them. So we want to make sure that we don't have anything in our process that's going to have the remote chance of introducing any adventitious contamination into the manufacturing process. Because we're dealing with patients who are extremely sick, sometimes the patient has pre-existing microbial contamination in their blood already. So our hope is that when we receive their blood, we have the necessary controls in place to make sure that that contamination doesn't proliferate and actually kill the patient's cells, because that creates a greater risk for the patient, because the patients are so sick at times that they can't even receive another apheresis to have their blood taken. And because of that, we want to have all the controls in place so that we don't introduce any adventitious contamination, such that we can get it back to the patient quick, fast, and in a hurry to save their lives. So next, I'm going to give a high-level overview of a manufacturing process for a CAR-T process. So we have our incoming leukapheresis. So the patient is at the clinic or the hospital. They get apheresis. Then we separate out their T cells, their white blood cells, and extract the T cells. Then we have the process of raw materials and consumables. Then there's always media involved in the manufacturing process. And then you have the actual manufacturing steps, and you have the final product. So again, these are the high-level buckets for CAR-T manufacturing, and these are the major buckets that we're going to discuss today in evaluating a risk-based approach for just-in-time release and a 2-day release of the product from a microbial contamination standpoint. So let's start with the leukapheresis product. So remember I said that this is the last option for patients, the CAR-T manufacturing. This process, they've normally gone through 3 other treatments, and the patient has received other treatments, and sometimes the patient is near death, and sometimes the patient either has an microbial infection that already exists, or when the leukapheresis process happens, the nurse or the doctor who are actually doing the process may cause contamination of the patient's blood. So from a risk-based standpoint, this is one of the most important steps in the manufacturing process of CAR-T, because if contamination occurs during this step, it's probably going to come into the manufacturing process, and those organisms that may be present from an improper leukapheresis will then get into the manufacturing process, and then we'll go through the growth phase or the incubation phase of expanding the cells, which is in a growth media that microorganisms love. That microorganism also has a chance to proliferate and cause the death of the patient cells. So this is a point in the step in the process where you want to make sure that you have some rapid release testing, because you want to be able to test the leukapheresis material just to ensure that there's no contamination up front coming into your manufacturing process. And this would be the step where you want to catch it, because at this step, the patient may still be available to get a second leukapheresis, and you don't want to introduce any contamination from the leukapheresis process into your manufacturing process. So I'm not going to read through each of these, but as you can see, these are high-level things that could happen at the leukapheresis process, and it also provides you with some mitigation actions that you can take at this step in the process to help prevent or mitigate potential microbial contamination from coming into your manufacturing process. And again, this is one of the more critical steps, because you want to catch it early, any potential contamination that's coming in from either an incorrect leukapheresis or from the actual patient material, because at this point you want to stop it from even being manufactured and getting into your manufacturing process. And I want to point out another fact here is that when you're performing this microbial testing to detect potential contamination, you want to have a test that can detect it quickly. So the traditional compendial test, which requires 14 days, would not be suitable in this case, because you want to be able to detect the contamination quick, fast, in a hurry, so that you can at least let the physicians or the clinicians know that, hey, this patient leukapheresis is contaminated and we need to get another sample before we can actually begin the manufacturing process, and hopefully that gives the patient a better chance of survival. So at the leukapheresis stage, these are some of the contributing factors, and some of the things that you should consider, considerations for a contamination control. So at this point, there's potential increased adventitious microbial contamination risk to the production process and the patient, because remember I said if you can detect the contamination before it actually gets into the manufacturing process, it's better, because you can then go get another apheresis from the patient and start the process again. And if the contaminated product does get into the manufacturing process, there's an increased risk to both the production process and the patient, because if a microbe is present and you then introduce it into the growth steps of the manufacturing process, that microbe is going to potentially proliferate, which is going to then compete for the same nutrients as the patient cells, and it's going to cause death of the patient cells, which means it's really not good for the patient, because you then have to go back and start the process again. So this is the stage in the process where you want to introduce some absence of contamination testing, because it provides important and valuable data, because if you can detect the contamination up front, you're not going to start your manufacturing process. You're not going to introduce an organism to your manufacturing process that you've not seen before. And typically in CAR-T manufacturing, when we see microorganisms that come in and we get a contamination, it's normally Pseudomonas, Salmonella, Staph epi, things that you would typically see of human nature. The most contamination that we would see in CAR-T manufacturing actually comes from the leukapheresis of the patient themselves and not from the manufacturing environment. So that's one key thing to keep in mind. And again, you want to consider performing testing at the leukapheresis centers to get it up front, or you either want to test it when you receive it at your manufacturing site. And again, because this is a very time-sensitive manufacturing process, both for the patient cells as well as for the patient life, you want to have a rapid method in place here, because you need to get your results back as soon as possible so that you can take action swiftly. So let's move to the next major bucket in the manufacturing of CAR-T products. So we have our leukapheresis material. So now let's consider all the raw materials and consumables that we're going to use in the actual manufacturing process. So in CAR-T manufacturing, we have to remember that all product contact materials are sterile, they're single-use, and they're disposable. So unlike traditional manufacturing where you have the bioreactors and the filling lines where the product actually comes into contact with those things, and you need the traditional cleaning validation, cleaning qualification, and effective cleaning program, in CAR-T manufacturing, everything is single-use, disposable, and is sterile, which minimizes the risk from a contamination control standpoint, because if you have a robust raw material program in place where you qualify your vendors, you've actually qualified your materials, this should be create a less opportunity for the ingress of adventitious microbial contamination. So at this point, if you have a robust raw material program in place, that is another step in your contamination control for CAR-T release. And again, I highlight the considerations that you should consider from a microbial contamination standpoint. The raw materials and consumables actually present a decreased adventitious microbial contamination risk to the production process and the patient because everything is sterile, it's single-use, and it's disposable. You will prevent potential adventitious microbial contamination. You will detect it upstream via your raw material program, which means that it will never come into contact with your patient material. So therefore, this is another additional step in your microbial contamination program. And in order for your raw materials program to be successful and to contain all of your controls that you need from a microbial standpoint, you need to make sure that when you bring these materials into the facility, they're going to come into the warehouse. And it's important to note for CAR-T materials, manufacturing materials, that a lot of the consumables that we receive, they're not triple bagged. So they're not the things that you will see in your typical manufacturing process. So you can either get a bag in and it just has one wrap on it, or you can get a bag and it comes directly in on a cardboard box. So you have to make sure that in order for your raw material program to be effective, even though they're single-use and sterile and disposable, remember that your production operators are going to be touching the outside of the bag. So you want to make sure that when you establish your cleaning and sanitization program that you take into account the raw materials that you're bringing into the facility, because you want to make sure that you physically remove all the dirt and the potential microbial organisms that are present on the exterior surfaces of those bags before they come into your manufacturing facility. And this is extremely critical for CAR-T manufacturing process because typically you're going to receive it in, you're going to unpack it, you're going to sanitize it, and then you're going to transfer it into the manufacturing facility. So you want to make sure that your cleaning procedures or your sanitization procedures are effective and robust, because otherwise you're going to be transferring exactly what came in on that material into your manufacturing facility. And I have a question from the audience. It says, is single use is also true for allogenic therapy? I think it is also true for allogenic therapy is that they also use sterile single-use disposable materials. So next, we're going to go into the next process in the manufacturing, the next major bucket. So when we say media in CAR-T manufacturing, we're talking about all those things that we add, the supplements that we add to allow for the patient cells to grow after we have made the transduction. So because these are aseptic manipulations, all critical aseptic manipulations in CAR-T manufacturing are performed typically in a DSC, which also meets the ISO 5 grade A standards. And we don't make media from scratch. We simply supplement media. We formulate it with sterile supplements. So all the media supplements are also sterile. When you're making your supplements inside of the DSC, you want to make sure that you have a risk-based EM program, including personnel monitoring. And most of the manipulations that are performed in the DSC for CAR-T manufacturers are short duration. And even though all the media components that we're adding together are sterile, as an additional contamination control, most CAR-T manufacturers will have an additional sterile filtration of their media. So again, these are all the things, again, that you would consider when you're thinking about your release strategy for CAR-T products, is that even when you perform activities in the grade A area, that all of these things are sterile. And because you do have personnel who are making sterile aseptic manipulations, you want to make sure that you're monitoring their gloved hands, because they're the closest thing to the process that you're going to get potential contamination from, is from the personnel gloved hands. And I failed to mention this at the beginning of the presentation, but in order to establish a process like this, you need to make sure that you have done a robust microbial risk contamination assessment. So you want to start from your incoming leukapheresis all the way to your final product, and you want to assess every step in your manufacturing batch record. And within each of those steps, you want to come up with potential contributing factors where microbial contamination may be introduced. And this is going to take a while to do, so you want to make sure that you have a cross-functional team assembled, and all the people, the SMEs, the people who have the knowledge of the process, and you want to make sure you have a microbiologist on board. You want to make sure that you have somebody on from your manufacturing science and technology group or your analytical science and technology group. You want to make sure you have somebody from facilities, from engineering, because you need to look at everything that's in place in your manufacturing area, and you want to make sure that you cover every area and every step in the manufacturing process based on the batch record where you think potential microbial contamination can be introduced into your process, as well as all those things that you need to do to mitigate those actions. And let's go and look at the considerations that we'll have for media. So again, because all the media is sterile and we're simply formulating sterile components together, and we have a terminal sterile filtration, the media process in CAR-T manufacturing also presents a decreased risk of adventitious microbial contamination. And again, you will have potential contamination detected upstream because all of these things are going to be a part of your raw material program. You will have qualified your supplier. You will have ensured that everything that comes in sterile is really sterile, and nothing with contamination will actually get into the manufacturing process. So at this point, you may not need to have any additional microbial testing. You would simply need to make sure that your filter integrity testing passes each time you add the media supplements together and filter them. So as you can see so far, all of this is building on the controls that you can have in place, because if you have all these controls in place during your actual manufacturing process, it really eliminates the need to have that feel-good testing at the end, and that's why I'm leaning towards the just-in-time release or the 2-day release, which I'm going to get into later. So now let's look at the manufacturing process. And again, I'm not going to read each of these, but as you can see that there are many buckets in the manufacturing process. And again, in the manufacturing process, every critical aseptic manipulation is performed within a biological safety cabinet, which meets ISO 5 grade A requirements. Most of the aseptic manipulations are of short duration. They're all performed in a controlled environment. Personnel have on the proper gowning. We have an environmental monitoring program in place to detect any potential contamination that the personnel may have had on their fingers or their hands while they're performing any of the manufacturing. And again, remember that most of these are just aseptic connections that you're making, and if they're the fluid pathway of the product is never exposed to the environment, and that's another control in CAR-T manufacturing. And wherever you think that your fluid pathway may be exposed to the environment, that's where you want to make sure that you're using a biological safety cabinet and you have all the environmental monitoring in there to help you to protect any potential contamination that you think may be present in your environment. However, if you do your microbial risk assessment and you map out every step in the manufacturing batch record, you will probably come to the same conclusion that your highest risk of microbial ingress will come from personnel gloved hands, because they're the ones who are making all the aseptic manipulations, and their fingers are the things that will come closest to the potential fluid pathway of the product and not the actual environment. So let's look at some microbial contamination considerations from the manufacturing standpoint. So again, the actual manufacturing process presents a decreased risk of the introduction of adventitious microbial contamination. Again, I will emphasize that it is all sterile single-use consumables that are utilized in the manufacturing process. There are no product contact surfaces that are not sterile and that are reusable in the process for CAR-T manufacturing. And again, based on that intense and very detailed microbial contamination risk assessment that you will perform, personnel gloved hands will probably come out as ranking the highest in your risk of potential sources of probable contamination into your manufacturing process. And finally, even though you shouldn't use this as a microbial contamination control strategy, but it's also reality that because we do use growth media in doing the cell expansion that support microbial growth, your process is really actually self-contaminating checking, because typically you have a minimum of 5 days of incubation in a nutrient-rich media that your patient cells are growing in, and if a microorganism is present, it's going to compete for the same nutrients in that media, and it's going to proliferate as well. And an example I can give of this is that one of my previous companies, we had a contamination that came in with the patient. However, the apheresis centers did not know that they had introduced a contamination. It was actually from a patient who had, they had to put the needle in the groin of the patient, and the patient had massive diarrhea, but the nurses thought that they had cleansed the area, sanitized, and disinfected the area well, so they assumed that there was no contamination when they sent the patient cells. However, 3 days of incubation of those patient cells, the entire bag turned black. And when we did the investigation, it was determined that the leukapheresis process had actually introduced salmonella during the leukapheresis process, and therefore the salmonella proliferated and it killed the patient cells, and therefore the patient had to be re-aphed again to get additional blood to start the manufacturing process. And again, this is one of those steps where it creates additional delay for the patient. And we don't want that to happen because the patient is already at their last point of life. So we really don't want anything to be introduced into the manufacturing process. So if we can catch the contamination up front before the manufacturing process starts, it gives greater hope for the patient because they can then get apheresis again and we can start the manufacturing process. I have 2 questions that have come in from the audience that I'm going to answer. The first question that says is, how often are you doing aseptic process simulations and what media is used? So for aseptic process simulations, again, you can take a risk-based approach. It depends on how your facility is designed. Because people are normally the equipment for cell therapy manufacturing, so your process is pretty much mimicked in every suite or room that you may have. So the equipment is the same, the people is the same, the process is the same. So I think it would depend on your company, but I think the agency is willing to accept that if you have like for like in all of your manufacturing areas and it's the same people who are performing the same manipulations in each of those suites, that you only have to do 1 aseptic manipulations for each of those suites, and you have to determine the frequency of how often you're going to repeat that aseptic process simulation. The media that is used for aseptic process simulations in CAR-T is the exact same media that's used in traditional pharmaceutical. It's triptychs or broth or any nutrient enhancement rich broth. So the next question is the just-in-time testing technically considered to be an in-process control or a lot release test? So in my opinion, the just-in-time testing is considered to be the lot release, because you're saying that you have demonstrated that your process is in such a state of control that you're not going to introduce any adventitious contamination throughout the process, and where you have decided to perform testing, all those testing results will come back as no growth, and therefore your process is in a state of control so that you don't have to perform that testing at the end of your process. But again, let me just reemphasize that you have to have performed a thorough microbial risk assessment. You have to have gone through each and every step in your manufacturing process and highlighted the risk and ensure that you have mitigating actions for all of those risks, and you have to have the testing in place up front to detect any potential contamination at those points that you deem are critical in your manufacturing process. So let's continue. These are some additional considerations. You have established procedures in place to prevent potential cross-contamination between patient lots. The potential increased risk of adventitious microbial contamination, again, at this point stems from the incoming leukapheresis and from the patient. And the greatest risk at this point will be delay of the product or no product getting back to the patient, which would be detrimental for the patient. And I have another question that just came in from the audience. Is there ever a time when the apheresis from the patient is received is found to be contaminated, but only slightly, and you continue to process, or do you start over no matter how much contamination? My suggestion would be that if you find contamination in the incoming material, you should ask to get a fresh material, because even if you identify the organism and you think it's a slow-growing organism, you never know how it's going to react when you introduce it into the growth phase of your manufacturing process. And you don't want to introduce any organisms to your facility that are not typically found in your facility. So that's the other concern when you actually bring in apheresis that's contaminated from the patient. You have now exposed your manufacturing facility to that organism as well, even though you should have controls in place so that it doesn't get out into your actual manufacturing facility. You just want to be careful that you don't put the patient at risk by continuing to process something that you know is contaminated, with the potential for it to proliferate and to kill the patient's cells. So the key in CAR T manufacturing is that we want to give a patient as much time as the patient needs to get the drug back to the patient so that the patient can recover, because every single day, every single minute matters for these patients, because they're literally at the point of death, and we don't want to do anything that's going to delay getting that product back to the patient. I also have another question from the audience. Does it make sense to keep the room classification as grade B area for manufacturing? I am assuming that this person is asking if the BSC has to be in a grade B or a grade C area, and if that's not the correct assumption, the person can let me know. But my recommendation would be to, based on your microbial risk assessment, to determine what actual room classification you need. So in CAR T manufacturing, there is nothing exposed in the area outside the grade B. So everything that you transfer from your warehouse that you sanitize, you transfer it into your grade D and then to your next higher classification. Nothing is open at that point. So everything you're doing is sanitizing and passing into the BSC. So in my professional opinion, I think that you should qualify the room as a grade B during your environmental performance qualification, but you should operate as a grade C based on the actual manufacturing steps that are occurring in that area. And if that doesn't answer the question, please let me know, and I'll try to answer when you respond. So let's talk about the final product. So again, everything that I've talked about so far is performed inside of classified areas. So the final product, you're pretty much adding in your cryoprotectant to protect the patient cells, because when you add the cryopreservation material to patient cells, it has the same effect on patient cells as it would have on the bacteria, that if you add it too fast, it will kill the patient's cells. It causes death to the patient's cells. So there's no way to prevent that. So you just have to have a validated process in place to ensure that when you're adding the cryoprotectant to your cells, that it's done in a way so that it doesn't kill the majority of the patient's cells. And the same thing will happen with bacteria. If bacteria is present, you add in the cryopreservation. You want to make sure that there's not going to be apoptosis for your bacterial cells as well. So again, the final product in cell therapy manufacturing, they consist of low-risk aseptic manipulations. Another thing I want to point out in CAR T manufacturing is that in traditional sterile manufacturing, the words open and closed are used a lot. However, when you try to transfer the word open and closed over into cell therapy manufacturing, there's no direct correlation, because in cell therapy manufacturing, for the most part, the fluid pathway of the product is never exposed to the environment. And what that means is that when you're working with the bags, when you make the connections, you typically clip off the two ends that you're going to make the sterile well, so that the patient blood never sees the actual environment. And that's why when you do your risk assessment, you're going to determine that the greatest risk is from the personnel gloved hands, because they're the ones who come closer to those aseptic manipulations that you're making. But the fluid pathway of the product is never exposed to the actual environment, even though we have all the controls in place to perform them in an ISO 5 grade A area. All the other rooms are classified because, again, it's all about risk mitigation. We want to make sure that we don't introduce any adventitious contamination, even though the risk of introducing any adventitious contamination is minimal. But again, you want to make sure that you're processing in an aseptic manner, because you want to make sure that you have taken all the steps that are needed to introduce controls to prevent any adventitious microbial contamination. So again, for the final product, the risk of introduction of adventitious contamination at this point is low, because you're really just making aseptic manipulations that have low risk. And when I say low risk, it just means that you're making sterile tubing connections and moving things from one bag to another. So again, I'll highlight some of the considerations from a microbial contamination standpoint. Again, there's a decreased risk at the final product stage of introducing adventitious contamination. You can perform endotoxin, Mycoplasma, and detection testing at this point if you like. These are all the traditional safety testing. Sorry, let me go back for a minute. I just want to point out one thing. So for endotoxin and mycoplasma, remember that in cell therapy manufacturing, there's no water in the process. So if you control your raw materials through testing up front and you don't have any way of introducing endotoxin and mycoplasma from your actual manufacturing environment, this is another thing that you should consider when you're doing your release of the product, is that if you control endotoxin and mycoplasma up front from your raw material program, there is a potential that you can eliminate these testing from the end, and that will also contribute to your just-in-time release, not only for the microorganism testing, but also for the other safety testing of endotoxin and mycoplasma. But again, you have to have a robust raw material program in place, and you have to demonstrate that those things are controlled to the point where you know that there's not going to be any endotoxin or mycoplasma introduced into your manufacturing environment. Just checking to see if we had another question. Nope. Okay. So in summary, I'm going to go with the two options that I provided from the beginning that I discussed. So option number one is just-in-time release, which means that at the end of your final fill of your product, and when I say final fill, remember this is for CAR T manufacturing, so it's not your typical 10,000 vials you would have for a traditional pharmaceutical. In CAR T manufacturing, every single patient lot, every single patient that we get is considered one lot. So it's 1 for 1. You get in one patient blood, that makes one lot. So the justification for just-in-time release is that the patient is waiting on the product. Remember I said this is the last treatment option for patients. This is life or death for them. So we really don't want to have anything that's going to interfere with getting this product back to the patient. You have the adequate engineering and procedures in place throughout the entire manufacturing process to mitigate any risk of microbial contamination. And again, remember I said that the highest risk of the introduction of adventitious contamination in CAR T manufacturing actually comes from the leukapheresis process and or the patient, and not from the manufacturing process. The majority of the manual aseptic manipulations in the manufacturing process present a minimal risk of adventitious microbial contamination. And if you have all the controls in place, the contamination will be readily detected upstream prior to patient release. So again, if you have done your risk assessment, you're performing testing in all the places that you think the contamination could be introduced, you have a robust raw material program in place where you're testing all your raw materials, you qualify your suppliers, you qualify your vendors, just-in-time release can be an option for CAR T manufacturing. However, if you're not willing to take the plunge and to go into just-in-time release of CAR T process, I am suggesting another option. So remember I said that all of the aseptic manipulations that are considered critical aseptic manipulations are performed inside of a biological safety cabinet. So if you consider those steps that are performed inside the biological safety cabinets being your critical steps where you could introduce adventitious contamination, and remember I also said that based on the risk assessment that the highest risk of the introduction comes from personnel gloved hands. So now if you get an ID of the flora of every organism that you recovered from those activities that performed in the BSC on those personnel gloved hands. So you want to consider the organism that you recover in the actual BSC, and you also want to consider those organisms that you recover on personnel gloved hands. If you're using a rapid detection method, the majority of those organisms are identified within 24 to 48 hours. So if you don't want to do just-in-time release, you can also reduce the number of days you perform testing by only focusing on those areas where you say that microbial potential microbial contamination can be introduced into your manufacturing process. I have another question that just came in. If the highest risk is the introduction of the leukapheresis sample, do you wait for bioburden results on the incoming samples before starting the process? So if you have a rapid method and you're focused on those organisms that are coming from a human, depending on which rapid method you use, you can have results in 1 to 2 days. So you should wait on those results before you start the manufacturing process. However, due to the criticality of the patient, most CAR T companies, they start the testing and they proceed with manufacturing at risk because the assumption is that the leukapheresis centers have not made any issues and that the patient does not have any pre-existing microbial contamination, although that can't be totally ruled out, but because of the criticality of trying to get the results, get the product back to the patient in a timely fashion, you start the test, but you don't wait for the results. However, if you start the manufacturing process and you do realize that your incoming material is contaminated and it's already in the growth phase of your manufacturing process and growth is not detected in your manufacturing phase, that's actually a good thing because in CAR T manufacturing, it's one of the things where you can actually, the doctors will actually request if you have an organism present in that patient lot to give it back to the patient because the benefits far outweigh the risk because they can give the patients antibiotics. They would pretty much want you to identify the organism and if you could do a minimum inhibitory concentration to see which antibiotics the organism is susceptible to, and the doctor will start that patient on that regime. So again, remember these patients are at the point of death, so the doctors really want to get the product back to them if possible, even if there's a contaminant present in the product. But you have to work with the regulatory bodies. Let me just put that caveat out there. You have to work with the regulatory bodies to get approval for releasing that product to the patient, and typically the regulatory bodies will give you approval in those instances because, again, the benefits far outweigh the risk for the patient in that regard. Okay. And I've already talked about this in my 2-day release option, is that if you focus on all the activities that are performed in the DSC, growth is detected usually within 24 to 48 hours, and the longest time for detection is typically an anaerobic organism, and you don't have anaerobic organisms because we don't have any anaerobic conditions in our manufacturing process. So you, again, when you're doing your method validation, if you're comparing it to the compendium method, you want to use the same organisms that are in the compendium method, but you also want to introduce organisms that are not typically in the compendium methods because in CAR T manufacturing, since you're dealing with human blood, you want to focus on those organisms that are going to be of clinical significance. So you want to use those in the validation of your rapid method as well, because those are the organisms that you're going to typically see, because remember I said that most of the contamination comes in with the patient leukapheresis materials, and they're going to be of clinical significance and not typical facility isolates. And again, those are all recovered typically within 2 days using a rapid method. So here I just have some examples of some rapid methods that you can use. Most CAR T facilities use the BAC-T, which is the one on the left. And there's also the BAC-TEC, which you can use, but most companies use the BAC-T because the BAC-T offers 2 incubation temperatures, whereas the BAC-TEC only offers 1 incubation temperature. So if the FDA has stated repeatedly that they want you to, even though this is a rapid method and they know it's advancing, they still want you, because the compendium method is still their gold standard. When you do your validation, they want you to compare it to compendial. And if you're comparing it to the compendium method, you have to have 2 incubation temperatures, 20, 25, and 30, 35. However, if your manufacturing process is that of cell therapy, where all of your manufacturing blood cells are incubated at 30, 35 degrees, you can then file your BLA to have only 1 incubation temperature for your blood product, because it's 30, 35. But during the validation, in comparison to compendium method, you have to do both temperatures. However, when you do your filing, you can submit the temperature where you perform your manufacturing activities, which is typically 30, 35 degrees, which is where you incubate your cells, and that's where you would expect your organisms to be recovered. Another thing I want to be careful of is that the FDA and other regulatory bodies have stated that they don't want to have a cookbook approach to telling companies what to use when you do your validations. For instance, how low do you go with your inoculum when you're doing your validation? You as a company need to determine your own inoculum level. But the FDA expects that if you have a rapid method that it's going to be, typically it's going to be more sensitive to the compendium method. So they want you to show that at least your rapid method can detect organisms at a higher sensitivity than that of the compendium method. So that's their only requirement, but they will not tell you exactly which inoculum level you have to use for your validation. I just wanted to point that out. And that's the end of my presentation. Thank you very much. And if you have any questions at the end, I'll be here after Laurie finishes her presentation. Stephen, I will turn it back over to you.
Thank you, Irving. Now, before I introduce our next presenter, Laurie Dahn, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen. And we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Laurie Dahn is the Director of Scientific Affairs at bioMérieux and has experience in clinical, environmental, and industrial microbiology. She is a technical expert on rapid and alternative methods and participates in the sourcing and evaluation of new technologies and potential partnerships in the field of microbial control. She provides scientific support to the healthcare business in North America and is responsible for managing feasibility testing and method development for bioMérieux instruments and culture media products. Laurie received her PhD from the University of Minnesota in microbial ecology. She also holds a master's degree in limnology from the U of MN and a bachelor's degree in the medical terminology from Ferris State University. I'll now be handing over to our second presenter. Welcome, Laurie.
Thank you, Stephen, and hello, everyone. Thank you, Irving, for your informative presentation. I think Irving provided a good overview of CAR-T product manufacturing and the importance of the product to the patient. I would like to say that the people I've met who work in cell and gene therapy are incredibly passionate, and they know that what they do saves lives. As Irving stated, they are many times a person's last chance of survival. I've also experienced that it's not uncommon when visiting a cell and gene therapy production site or at a conference for someone to provide a personal testimonial on how these products are saving the lives of their friends, relatives, or even themselves. So clearly there is a need to get these life-saving products in the right condition to the patient as quickly as possible. And Irving outlined two strategies, just-in-time and two-day release for CAR-T products using a risk-based approach. In my presentation, I'm going to focus on smart quality solutions for CAR-T product release, specifically focusing on automated microbial detection and Mycoplasma testing. But before I start, I would like to provide a little background on who we are. bioMérieux was created by Dr. Alain Mérieux in 1963, and he is the grandson of Marcel Mérieux, who was a student of Louis Pasteur. And Marcel Mérieux founded the Institut Mérieux in Lyon, France in 1897, where he developed the first anti-tetanus sera. So today, Marcel's great-grandson, Alexandre Mérieux, continues the family tradition and is our current chairman and CEO. This slide highlights our commitment to diagnostic microbiology. bioMérieux provides the widest portfolio for efficient quality control solutions. So these include environmental monitoring, microbial identification, standardized strains with our very popular BioBall, endotoxin testing using recombinant factor C, validation services, and lastly, what I will be focusing this presentation on is sterility and Mycoplasma testing. Now, Irving provided an overview of CAR-T production, and This slide is showing a highly simplified needle-to-needle supply chain cycle. We know that there are numerous challenges associated with manufacturing cell and gene therapy products, which I'll summarize shortly. And for these products, the supply chain activities must be highly synchronized and managed to successfully deliver the right product to the right patient in the right condition. And again, all of the testing and the manufacturing for these products are to ensure patient safety. I would like to highlight the differences between allogenic and autologous, specifically from a manufacturing point of view. The differences highlight the need for enhanced chain of custody and chain of identity and the need for smart quality solutions. So for allogenic products, one batch serves many patients, and the product may also be cryogenically stored for weeks to months. Sometimes this is called a scale up. However, for autologous products, one batch serves one patient. And so this seems easy when you're managing one product or even 10 products. But what if each of these individuals were to equal 100 products? How would you manage 300 products or even 700 products at a time? Therefore, the biggest challenge that I hear when I talk to someone in cell and gene therapy is the need for chain of custody, data integrity, and electronic tracking. Now, I've been at several conferences, and what I've heard from the FDA themselves is that they've stated many times that they focus at where there are risks for cross-contamination. Therefore, when you're performing your quality testing, it's important to use closed systems and test methods that provide the best data integrity. So just to summarize some of the challenges of cell and gene therapy, these products usually have a very low production volume and short shelf life. It can be just a couple weeks or maybe only a few days. They're sensitive to temperature, and there may be a need to maintain a cold supply chain. The raw materials are complex, and in fact the starting material itself is highly variable. The analytical methods used for testing are highly complex, and you need enhanced tracking and data integrity, and you have to have the ability to scale up. So the importance of managing these challenges is again, to get the right product to the right patient in the right condition. So coming back to our cell therapy production process, an additional challenge to quality testing is that it is a continuous process and takes days, not months. So a few examples from a recent conference showed a typical production cycle of 14 to 20 days. So the goal, again, is to get the cells back into the patient as soon as possible. And these products cannot be terminally sterilized, and they're processed following aseptic techniques. So there is no time for end product testing following traditional methods, but rather a risk-based approach is used, similar to what Irving Ford described in the previous presentation. Therefore, a robust quality management system must be established using smart quality solutions. An additional challenge is that the majority of testing is performed in a separate centralized QC laboratory. There's a lot of value in finding opportunities for rapid testing and bringing this testing out of the lab and directly to the production floor. And the best way for this to happen is to work with suppliers that offer rapid solutions. Now, in my presentation, I'm going to focus on two rapid testing solutions that are ideal for cell and gene therapy. And the important thing is that they are simple to use, fast, and accurate. In Irving's presentation, he highlighted the use of ATMP guidelines, and here I describe the three main types of rapid microbial methods that can decrease the time to result from the 14-day compendial test that's described in both the EP and USP. Now, alternative methods have been embraced, and they're described in EP 516 and USP 1223, and they include growth-based methods such as CO2 production and bioluminescence, direct measurement such as solid phase cytometry and flow cytometry, as well as cellular component analysis. More recently, the EP has published 2627 to address microbiological examination of cell-based preparations that also includes the use of rapid methods. There's also a new chapter, USP 1071, that was recently published in June of 2019 and will become official in December of 2019 for rapid testing of short-life products using a risk-based approach. So what I've seen really in the past 5 to 10 years is that there's really been a shift from rapid methods being an alternative to the compendial test to the compendial test being a rapid method. So rapid methods can reduce the time to detection from 14 days to as little as 4 hours. So one growth-based rapid method technology based on CO2 production that is ideal for cell and gene therapy products is the BacT/Alert 3D microbial detection system. The instrument is modular. For example, here is the BacT/Alert 3D combo. So it combines all of the BacT/Alert 3D capabilities in a compact format. It includes a control and incubation module in a single instrument, and it has a total incubation capacity of 120 cells. Here is shown the BacT/Alert 3D 240 system that consists of a control module on the left and one incubation module on the right that is made up of 4 incubation drawers of 60 cells, giving a total capacity of 240 cells. So the control module can actually enable up to 6 incubation modules, so providing that flexibility. So therefore, the full instrument configuration can allow incubation of 1,440 bottles. Now, as Irving mentioned in his last presentation, the flexibility and modularity continues with the BacT/Alert 3D Dual T system. Now, this configuration has both a low and high temperature incubation modules, allowing it to closely follow the Pharmacopoeia guidelines. So the low temperature module allows for incubation at 20 to 25 degrees, and the high incubation module allows incubation at 30 to 35 degrees. So similar to the single temperature modules, additional incubation modules can be added. Now, with the BacT/Alert system, the only reagents that are needed are the aerobic and anaerobic bottles that have unique barcodes. So once the sample is added to the bottle, you have full data integrity. The BacT/Alert system utilizes a colorimetric sensor and reflected light to monitor the presence and production of CO2 dissolved in the culture medium. So once bottles are loaded, the colorimetric sensors are scanned every 10 minutes. Microorganisms produce CO2 as they metabolize the substrates in the culture media, and this changes the color of the gas-permeable sensor in the bottom of each culture bottle from blue-green to yellow. So once growth is detected, the system alarms both audibly and visually, and the sample data is recorded. Now, the testing of the BacT/Alert system is incredibly easy. It's just 3 steps. You inoculate the media bottle, then you scan the bottle, and then you load them into the incubator. I've talked to a lot of cell therapy companies, and this is what they love about the system. They love that it is so easy. So there's no worry about removing enriched sample or performing additional manipulations or testing that may include reagent management. So again, where there's a high need for chain of custody and chain of identity and data integrity, you want to use a smart quality system like the BacT/Alert 3D. Now, the BacT/Alert system is an automated culture method. The bottles are continuously monitored, reading the bottle sensor every 10 minutes. The continuous monitoring allows for faster detection of positive results, and the reading is objective and completely independent of turbidity, so there's no need for subculture. The Dual T system allows for incubation following the harmonized Pharmacopoeia guidelines and is 21 CFR compliant. Now, there have been numerous publications using the BacT/Alert system for traditional pharma as well as cell and gene therapy product testing. And recently, the NIH published in the, oops, let me go back one, I'm sorry. Recently, the NIH published in the Journal of Clinical Micro in February of 2019, comparing the traditional and automated culture systems on 118 microorganisms. This is probably the most comprehensive study that I've seen, and the results showed that the BacT/Alert system can be used as an alternate sterility test. Now, with that said, most cell and gene therapy companies using a risk-based approach are focusing on clinically significant microorganisms that are usually detected within 2 to 3 days. And as Irving stated, their contamination is being detected within 24 to 48 hours. So now I'd like to shift to our second rapid microbial method, and that is Mycoplasma. So just to give everyone an overview of the challenges of this microorganism, it is ubiquitous. The cell cultures provide a very favorable environment for their growth. And if you have a contamination event, the quality and quantity of the product is decreased, and some of the species are clinically significant. Mycoplasma is difficult to prevent, it's difficult to detect, and it's very difficult to eliminate if you have a contamination in your manufacturing facility. So especially for cell and gene therapy, there is a need for a fast, simple, and robust solution. So in cell and gene therapy, the traditional method really cannot be performed due to the long testing time. This slide shows the traditional method that can take 28 days for results. Here's presented the indicator cell culture method, and while it's faster than the culture method, it still takes 5 to 8 days and requires a lot of technical expertise. Now, there are alternative molecular tests, and I think most cell and gene therapy companies are using molecular test systems. They're available to provide results in about 5 hours. But the test method requires expertise, and many times a molecular biologist is needed. The test is prone to contamination due to the open test system, and a specialized laboratory is required. So at this point, I mean, what if I could tell you that you could take a 28-day traditional test or even a 5-hour nucleic acid test that requires a high level of expertise and turn it into an hour and make it so simple that anyone can perform it. It doesn't require a special lab, and it could be potentially performed directly on the production floor. So what I would like to introduce is the molecular lab in a pouch for testing Mycoplasma. This is the Biofire Mycoplasma assay, and it's the fastest, easiest, and simplest Mycoplasma test method available. The test is a completely closed system for performing molecular Mycoplasma testing and takes less than 2 minutes of hands-on time and provides results in about an hour. It's so simple, it can be performed by anyone, anywhere, and at any time. The Biofire system consists of 2 components, the film array instrument and the single-use molecular lab in a pouch disposable. So just with these 2 items, you have everything that you need to perform fast, accurate Mycoplasma testing. So the compact film array instrument performs the extraction, amplification, and detection in a single machine. You can actually connect up to 8 instruments together for higher throughput. Now, we've completely transformed the Mycoplasma analysis from something very complex to something very, very simple. So this pouch contains everything to realize a molecular analysis from sample to result. So the sample is injected and goes from the lysis chamber to detection chips and includes 14 PCR assays designed to detect more than 120 Mycoplasma species. It contains all the controls necessary to control each step of the reaction. The reagent pouch system also decreases waste because 1 pouch equals 1 test, and it simplifies inventory in that the pouch itself is room temperature stable. So just giving an overview of the steps, similar to the BacT/Alert, that it's something very simple to do. the workflow for the Biofire Mycoplasma assay is also simple. You simply inject the hydration solution and unprocessed sample into the pouch. You place the pouch into the instrument and start the run, and the software reports whether or not the mycoplasma is detected. All this happens in less than an hour. You have easy-to-interpret results. The report that's generated shows that you either have mycoplasma detected or mycoplasma not detected. So again, comparing the different methods, the traditional method for cell and gene therapy is really not feasible at all to perform since it takes 28 days. So just looking at other nucleic acid methods compared to the Biofire method, you can see that the Biofire method shows a significant reduction in time. A novice can perform the assay. You have low contamination risk because it is a closed system, and the reagents can be stored room temperature and can be performed anywhere. So just to summarize the new Biofire Mycoplasma assay, the test is easy. Again, there's no PCR skills that are needed. There's no special lab that's required, no precise measuring or pipetting. You have very minimal data entry. It's automated, and it provides simple standardized results. It's easy. It only requires 2 minutes of hands-on time, and the sample to result in less than 60 minutes. And finally, it's comprehensive. It can detect more than 120 strains of Mycoplasma and molecules. You can test raw material, in-process, and final product. It's 21 CFR Part 11 software, and we provide full validation support. So the benefits of implementing a rapid contamination test, such as the BacT-Alert and the new Biofire Mycoplasma, includes the use of automation and improved work efficiency and data integrity, but it also provides standardization, ease of use, and objectivity. So both solutions are easy, and they can be brought out of the lab and close to manufacturing where they are needed. So to summarize, cell and gene therapy production is highly complex with numerous challenges. There's a need for enhanced chain of custody and data integrity to ensure the right product is delivered to the right patient without cross-contamination. New regulations allow a risk-based approach for microbial contamination and mycoplasma testing, and new alternative methods are moving the lab to the production floor. So with that, I would just like to thank Brian Whitehouse and Marianne Shu for providing some of my content today. This is my contact information if any of you have questions about the BacT-Alert or the Mycoplasma assay. And with that, I will hand this over back to Stephen. Thank you.
Thank you, Laurie, for that great presentation. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the Questions tab located directly below your webinar screen. So now our first question will be for Irving, and our first question is, what is the biggest manufacturing challenge for CAR-T products?
The biggest challenge is hiring personnel, especially in the area where we are in New Jersey. There are about 5 CAR-T manufacturing companies here, so we're all competing for the same resources, and that's one of the biggest challenges. The other challenge is that because one patient does equal one lot, and there's lots of variability in the patient cells, it's very difficult to know if a patient lot, they don't act the same, so when you're manufacturing, there's just a lot of variation in the process, so it's never a standardized process. So I always tell people who are going to work in cell therapy to forget your typical manufacturing life, and you have to come into a mindset, because everything in cell therapy is gray. It's no longer black or white, and you have to be able to make decisions instantaneously on the floor, and investigations can't take the typical 30 days. You have to have a triage right away. because the patient is waiting and the cells are really time sensitive. So you have to have a mindset so you're quick thinker on your feet, because you have to make a decision, live with that decision, and keep the process moving.
Thank you. Our next question is, are the regulatory bodies on board with such risk-based approaches? I believe that again is for you, Irving.
Yes, that is for me. So I was just at the PDA Global Micro Conference, and I was speaking to some of the reviewers and lead investigators, and they actually asked me for my presentation because they said they actually like the fact that companies are now taking a holistic and comprehensive approach and actually mapping out the process from a risk-based standpoint. So they're actually on board with it. I don't know if they are ready to jump into the whole just-in-time release, but they're definitely on board with the reduced timeline at the end. But I think as more cell therapy companies come on board, and the more important it is to get the product back to the patient in a hurry, and the more that this becomes the standard treatment for people instead of the final treatment, that the regulatory bodies are going to be more accepting. I think it's a matter of the company defending their process or their stance and making sure that they have the data and the risk assessments to support it.
Thank you. Our next question is, with the backed alert system, does bioMérieux have media available that allows neutralization of antibiotics? And that'll be for you, Laurie.
Yes. Yeah, good question. The media that we have available, we have standard bottles for both aerobic and anaerobic, and we also do provide resin bottles. And the resin bottles are designed to specifically neutralize antibiotics. So the answer is yes, we do have media specifically for neutralization of antibiotics.
Thank you. And our next question is, when will the Biofire Mycoplasma test be commercially available? That'll be again for you, Laurie.
Oh, yeah. Currently, the test is in pre-market customer testing. We anticipate the launch for commercial availability in the U.S. in March of 2020.
Fantastic. Moving on, our next question is, how important are rapid methods to CAR-T manufacturing? And that'll be for you, Irving.
They're extremely important. Actually, the regulatory bodies, that's one of their key questions when they're doing inspection of CAR-T facilities is, how can you prove to us that based on the number of patients that you're going to get, that you can actually manufacture them and get them back to the patient in a timely manner? And as a result of that question, they do expect you to have rapid methods, because the traditional 28-day Mycoplasma, 14-day sterility, they do not fit into the whole CAR-T platform, because the manufacturing process for CAR-T product is typically only 10 days. So you can imagine if you have a process that the patient is dying and they're waiting for 10 days to get their product, you don't want to delay it another 14 days for waiting on the result. That's really not going to provide value. So they really expect you to have rapid methods when you file an application for a cell therapy product.
Thank you. Our next question is for Laurie. Our next question is, does the Biofire Mycoplasma identify the species of Mycoplasma?
Yeah, we get this question quite a bit. The assay was actually designed as a presence-absence test. So it doesn't identify the specific Mycoplasma species, but rather Mycoplasma detected or Mycoplasma not detected.
Thank you. And with that, we will actually conclude the questions and answer session. But thank you, audience members. Oh,
Stephen, I had a question that came in that I don't think we gave an answer to. I just want to make sure that we answered it.
No problem.
What is an acceptable endotoxin specification for cell therapy products? Is it expressed at EU per mil or EU per kilogram per hour? It is actually expressed at EU per mil, and you would typically use your average body weight of the patient population to determine the endotoxin calculation that you would typically do for standard pharmaceutical products.
Perfect. I'm sorry for missing that question. Great.
Oh, no, that's okay.
And with that, we will conclude the question and answer session. Before we finish the webinar, I'd like to ask our presenters if you have any closing remarks for our audience. So, Irving, is there anything you'd like to let our audience know about?
Yeah, I would just like to thank the audience for actually participating today in the webinar. And CAR-T is an exciting world, and if you're not working in CAR-T, I would greatly encourage you to get into the CAR-T field, because I believe that personalized medicine is going to be the way of the future, and CAR T manufacturing is never a dull moment or a boring moment, and I'm really proud to say that I work in a manufacturing environment where I actually can see the results going back to the patient, and I'm actually saving patient lives, and I can actually see the results of changing people's lives. So I'm very proud to work in CAR T, and again, thanks everyone for calling in today.
Thank you. And Laurie, do you have any closing remarks for our audience today?
Yeah, I agree with Irving. Thank you for your comment. It's a very exciting area, and I'm very interested to see how things will be evolving with the guidelines, especially for the risk-based approach. The just-in-time and the 2-day release, I think, makes sense, providing, risk. That's the big word to use for CAR T production is a risk-based approach.
Great. And I would like to take this opportunity to thank our presenters. Once again, that was Irving Ford, head of the CAR T QC Laboratories at Celgene, and Laurie Dahn, Pharma Microbiology Scientific Director at Biopharmicoplasma. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.